So Chargaff's rule. The Chargaff's rules, it means that the DNA has equal amount of adenine and thymine residues because adenine is usually paired with thymine. Adenine pair with thymine. So whenever in DNA you have adenine and thymine, so adenine is usually found to pair with thymine through a hydrogen bond. And in adenine and thymine, we have only 2 hydrogen bonding. So also there is an equal amount of guanine and cytosine because guanine is also pairing with cytosine. And this pairing is through 3 hydrogen bonds. So between guanine and cytosine, there is a pairing, and the pairing is through hydrogen bond. So for the adenine and thymine, there are 2 hydrogen bonds. For the guanine and cytosine, there are 3 hydrogen bonds. So this is called as Chargaff's rules of molar equivalence of between purines and pyrimidines in DNA structure. So it's just trying to give us an idea of how these nucleotide bases are pairing. So it's the equal amount of adenine is also equal amount of thymine because thymine is pairing with adenine. So if you have 1000 adenine in DNA, then you have 1000 thymine. If you have 200 guanine in DNA, it means that you have 200 cytosine because they are pairing with one another. That is why the DNA is double helix. But RNAs, which are usually single-stranded, they are single-stranded, so they do not obey Chargaff's rule. Remember that I told you that DNA is double-stranded, while RNA is a single-stranded. So as a result of that, the Chargaff's rule will not apply to RNA molecule. So this is how it is. Adenine pair with thymine. So we have 2 hydrogen bond. This is hydrogen bond, and this is also another hydrogen bond between adenine and thymine. Then for the cytosine and guanine, we have 1 hydrogen bond here, another one here, then another one here. That is what makes it to be 3 hydrogen bond. The DNA double helix. The double helix structure was proposed by Watson and Crick, Francis Crick and Watson. So Watson and Crick, they are the scientists that proposed the double helix structure of DNA in the year 1953. And the DNA is right-handed double helix. So it is right-handed. When you say it is right-handed, it means that it is moving in spiral through right direction, and it consists of 2 polydeoxyribonucleotide chain twisted around each other on a common axis of symmetry. Then the chain are fair in antiparallel manner, and in an antiparallel manner, the 5' end of one strand is paired with the 3' end of the other strand. So the complementary strand. The 2 strands of DNA are not identical, but the 2 strands are complementary to each other. They are not identical, but they are complementary to one another. If in one strand you have adenine, then the other strand you have thymine, then two hydrogen bonding. Then also at the same time, guanine paired with cytosine. That is, if you have guanine in one strand, then the other strand will have cytosine, and is through 3 hydrogen bonding. So ladies and gentlemen, this is where we are going to stop for this DNA. I think